Okay students, today class we are going to discussing translation. In this translation, before translation I can ask the few questions. What are the questions I have to ask means? Terminator codons are 3. UAA, UAG, UGA. Sometimes same sequence means what? UAA, UAG, UGA sequence anticodons also may be present. But where does anticodons are there? TRNA. Suppose this one TRNA is there. According to your textbook diagram, itara kottiru TRNA. Correct? This TRNA mele UAA anticodon is there. Anta tilkodi. This anticodon TRNA brings which amino acid? Which amino acid they have to bring? I am saying anticodon. I am not saying codon. Okay now. So UAA anticodon is there means what is the codon of UAA? A, U. Correct? This is codon. This is called codon. Where does codon is present? MRNA. Correct? So MRNA mele AUU codon idre adike anticodon yavadu? UAA. Yes, okay na. AU is code which amino acid? Isoleucine. This is important. Correct students, okay na? Anticodon does not code any amino acid students. Codon will be code amino acid. TRNA mele anticodons are there. TRNA mele UAA and the sequence ide. Idu yavu amino acid ne tavamaralla anta ankondanದ್ರೆ ಅದು wrong. Correct students, okay na? So UAA is completely brings that one. Which amino acid? Which one? Isoleucine. Henge identify ಮಾಡಬೇಕು ಗೊತ್ತಲ್ವಾ? ಓಕೆ ನಾ? A first identify ಮಾಡಬೇಕು. UCA. ಕರೆಕ್ಟಾ? ಓಕೆ ನಾ? ಸರಿ ಇದಲ್ಲ. ಓಕೆ ನಾ? ಇದು U ಅಂದ್ರೆ ಫಸ್ಟ್ ರೋ ನಲ್ಲಿ ಬರುತ್ತಲ್ಲ. ಫಸ್ಟ್ ರೋ ನಲ್ಲಿ which horizontal box? Third horizontal box ನಲ್ಲಿ ಬರುತ್ತೆ. Third horizontal box. Okay? So that is called AUU. Okay? What is the identification mark of third box? Third box identification mark 26314. Correct? But identification mark means what I given? PLSD illustrated mass values. Correct? ಅದೇ ಬರುತ್ತಲ್ವಾ? mass ಬರುತ್ತೆ ಪ್ಲಸ್ illustrate ಅಂದ್ರೆ isoleucine ಬರುತ್ತೆ ಪ್ಲಸ್ ಮಿಥಿಯೋನಿನ್ ಬರುತ್ತೆ. ಬಟ್ ಮಿಥಿಯೋನಿನ್ ಮಾತ್ರ ಲಾಸ್ಟ್. AUG ಲಾಸ್ಟ್ ಬರುತ್ತೆ. ಕರೆಕ್ಟಾ? ಮಿಕ್ಕಿದೆಲ್ಲ 3 ಕೋಡಾನ್ಸ್ ಇದೆಲ್ಲ 3 ಪ್ಲಸ್ ಮೇಲೆ ಇದೆ 4 how many 6 okay na totally 6 codons in isoleucine. Okay na like this. How many codons for isoleucine? sorry 3 codons only for andre third horizontal box. Correct? Third horizontal box nalli first box nalli ella first 3 are code 1 amino acid, last 1 codon is code 1 amino acid methionine. Okay, and today we are discussing about TRNA. TRNA is also called which one? The adapter RNA, or it is also called which one? The soluble RNA. Students, it is also called soluble RNA. Why it is called adapter RNA, students? Because they can adapt the amino acids. Why it is called soluble? Under they can attach with the amino acid. They are attached with the amino acid and they are soluble or in the amino acid or not. That is the meaning. So soluble RNA, adapter RNA. And the 2 more names are there for transfer RNA. This transfer RNA, it is the smallest RNA anta helbodu. Smallest RNA. Around 20 different plus type of the tRNAs are there in the cytoplasm. Okay? But these tRNAs, come on, in the 2-dimension structure. So it is okay, or it is also called them as secondary structure. The secondary structure of the tRNA, it looks like a which shape? Clover leaf shape. Adike it is called clover leaf model anta karitam. Ade tertiary structure anta karidivi, or it is also called 3D picture anta helbodra. 3-dimensionally it looks like a which one? Inverted L shaped structure. Inverted L shaped, very important students. In textbook it is mentioned properly, inverted L shaped. How it is inverted L shaped means it is completely, you see, this is about the inverted L shaped structure. Inverted L shaped structure, same, it is about the 5 prime, 3 prime, it will be showing more coiling like this. But in the secondary structure, it will be look like a clover leaf model like this. This is clover leaf model. Now we can discuss clover leaf model only. But all TRNA same structure. But secondary structure nalli one type tara kanisbodu. Tertiary structure nalli innond tara kanisbodu. But all are having the same structure. Correct? Okay? But shape matra change aagbodu. Secondary nalli, tertiary nalli. Okay, now clover leaf model. This clover leaf model is commonly consist of 5 major arms are there. 5 major arms are there, and idralli 4 loops are there. Yen sara 4 major arms are there andre, sir 5 major arms andre. The first one is called acceptor arm. Acceptor arm. Second one is called the mass anticodon arm. Anticodon arm. Third one is called TCC arm. Next one, variable arm. Variable arm. Last one is called DHU arm. 5 different arms are there, students. We have to discuss about acceptor arm. Okay. Sir, RNA andre single stranded, but it looks like a double stranded. But actually single stranded. You see, are you observing, students? This is 5 prime polarity. This is 3 prime polarity. But why sir RNA ge shape bandide? Linear ಆಗಿರಬಹುದಲ್ಲ mRNA ಬಗ್ಗೆ ಹೇಳುವಾಗ. mRNA ತರ linear ಆಗಿರಬಹುದಲ್ವಾ? ಯಾಕೆ ಇದು ಅಂದ್ರೆ because of base pairing, complementary base pairing. Suppose adenine uracil is there, here adenine is there, uracil is means easily bind. Suppose here guanine and cytosine is there, means what is? Same like that one. Here also cytosine, guanine, here also okay na cytosine ide andre guanine, like that one. But ಯಾಕೆ ಸರ್ ಇಲ್ಲಿ ಪೇರ್ ಆಗಿಲ್ಲ ಅಂದ್ರೆ here unusual means what? Okay na non complementary base pairs are there. Non complementary base pairs, ಅದಕ್ಕೆ they can form that one. This is called the as a loop anta ಕರಿತೀರಾ, and this is called stem anta ಕರಿತೀರಾ. Stem ಅಂದ್ರೆ ಏನು? paid part ರಾ. ಈ ತರ pairing ಆಯಿತು ಅಂದ್ರೆ that is called stem ಅಂತ ಕರಿತೀವಿ. unpaid part ಗೆ ಏನು ಹೆಸರಪ್ಪ? loop ಅಂತ ಕರಿತೀರಾ. Okay, so here stem ಮಾತ್ರ ಇರುತ್ತರ. There is no loop in the which area. This is called acceptor arm ಅಂತ ಕರೀತೀರಾ. This is called acceptor arm. ಯಾಕೆ ಸರ್ ಈ ರೀಜನ್ಗೆ ಆಕ್ಸೆಪ್ಟಾರ್ ಆರ್ಮ್ ಅಂದರೆ? ಎಸ್, ಇದೇ ರೀಜನ್ನಲ್ಲಿ ಅಮೈನೋ ಆಸಿಡ್ ಬೈಂಡಿಂಗ್ ಆಗುತ್ತಾರೆ. ಅದಕ್ಕೆ ಇಟ್ ಈಸ್ ಆಲ್ಸೋ ಕಾಲ್ ಅಮೈನೋ ಆಸಿಡ್ ಬೈಂಡಿಂಗ್ ಸೈಟ್. ಅಮೈನೋ ಆಸಿಡ್ ಬೈಂಡಿಂಗ್ ಸೈಟ್. ಅಮೈನೋ ಆಸಿಡ್ ಬೈಂಡಿಂಗ್ ಸೈಟ್. ಓಕೆ, now you have to speak out. ಇದು 5 prime polarity of RNA. ಇದು 3 prime polarity of RNA. Where does free OH group is there? 3 prime. That's why you can guess that one. Where does amino acid can be bind? 3 prime end only. Why? Because OH group is there. Here only amino acid can be bind. Correct? tRNA function. What is the function of tRNA? They can bring the amino acid. Before bringing, they should attach with the amino acid or not? ಮತ್ತು ಅಮೈನೋ ಆಸಿಡ್ ಅಟ್ಯಾಚ್ ಆಗಬೇಕಂದ್ರೆ TRNA ಮೇಲೆ ಒಂದು ಬೈಂಡಿಂಗ್ ಸೈಟ್ ಇರಬೇಕು. ಅದಕ್ಕೆ ಯಾವ ಬೈಂಡಿಂಗ್ ಸೈಟ್? ಅಮೈನೋ ಆಸಿಡ್ ಬೈಂಡಿಂಗ್ ಸೈಟ್ ಅಂತ ಕರಿತೀವಿ. ಅರ್ಥ ಆಯ್ತಾ? ಸೊ ಯಾವ ಸೈಟ್ಗೆ ಅಟ್ಯಾಚ್ ಆಗುತ್ತೋ ಅದಕ್ಕೆ ಅಮೈನೋ ಆಸಿಡ್ ಬೈಂಡಿಂಗ್ ಸೈಟ್, ಆರ್ ಇಟ್ ಇಸ್ ಆಲ್ಸೋ ಕಾಲ್ ಆಕ್ಸೆಪ್ಟಾರ್ ಆರ್ಮ್ ಅಂತ ಕರಿತೀವಿ. ಆಕ್ಸೆಪ್ಟಾರ್ ಆರ್ಮ್ ಇಟ್ ಈಸ್ ಕಂಪ್ಲೀಟ್ಲಿ ಪ್ರೆಸೆಂಟ್ ಇನ್ ದ 3 prime polarity ಆರ್ 5 prime? 3 prime. ಆರ್ ಯು ಅಬ್ಸರ್ವಿಂಗ್ ಸ್ಟೂಡೆಂಟ್ಸ್? 5 prime polarity ಕಂಪೇರ್ ಮಾಡಿದ್ರೆ 3 prime ಸ್ವಲ್ಪ ಬೇಸಸ್ಸು ಎಕ್ಸ್ಟ್ರಾ ಇದೆ ನೋಡಿ. ಆ 3 nitrogenous bases, unpaired nitrogenous bases are there. ಅದೇ CCA ಅಂತ ಕರೀತಾರೆ. cytidyllic acid, adenyllic acid ಅಂತ 3 unpaired nitrogenous bases we can observe in the acceptor arm. ಅದಕ್ಕೆ acceptor armಗೆ CCA end ಅಂತನೂ ಕರಿಬಹುದು ಸ್ಟೂಡೆಂಟ್ಸ್. CCA end ಇದು ಇಂಪಾರ್ಟೆಂಟ್ ಸ್ಟೂಡೆಂಟ್ಸ್. CCA end. CCA ಅಂದರೆ 3 unpaired nitrogen bases. where it is present ಅಂದರೆ acceptor arm. ಈ ಆಕ್ಸೆಪ್ಟಾರ್ ಆರ್ಮ್ಗೆ ಅಮೈನೋ ಆಸಿಡ್ ಬೈಂಡ್ ಆಗುತ್ತೆ. Then we can go for acceptor arm is finished. Acceptor arm is providing the binding site for provide the binding site for what? Amino acid binding site. Amino acid binding ಆಗಬೇಕಂದ್ರೆ it is completely helpful. Correct? ಈಗ next one, anticodon arm. ಆಪೋಸಿಟ್ ಟು ದ ಆಕ್ಸೆಪ್ಟಾರ್ ಆರ್ಮ್ ನೋಡ್ತಾ ಇದ್ರಾ, ಒನ್ ಆಫ್ ದಿ ಆಂಟಿ ಕೋಡಾನ್ ಆರ್ಮ್ ಈಸ್ ದೇರ್. ಈ ಆಂಟಿ ಕೋಡಾನ್ ಆರ್ಮ್ ಆಲ್ಸೋ ಇಟ್ ಕಂಟೈನ್ ದ ಟೂ ಪಾರ್ಟ್ಸ್. ಒನ್ ಈಸ್ ಸ್ಟೆಮ್, ಸೆಕೆಂಡ್ ಒನ್ ಈಸ್ ಕಾಲ್ ಲೂಪ್. ಸ್ಟೆಮ್ ಅಂದ್ರೆ ಗೊತ್ತಾಯ್ತಲ್ಲ, ಸ್ಟೆಮ್ ಅಂದ್ರಿಂದ ಪೇಯ್ಡ್ ನೈಟ್ರೋಜನಸ್ ಬೇಸಿಸ್ ಅಂದ್ರೆ ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಬೇಸಿಸ್ ಆರ್ ದೇರ್, ಬಟ್ ಹಿಯರ್ ಅನ್ಪೇಯ್ಡ್ ನೈಟ್ರೋಜನ್ ಬೇಸ್ ಆರ್ ದೇರ್. But very very important, 3 unpaired nitrogen bases is present in the which loop? This is called anticodon arm anta karitivi anta telida. Ee anticodon arm nalli command 3 unpaired which bases are there? Nitrogen bases are present. Nitrogen bases are present called anticodons of mRNA. mRNA ಮೇಲೆ ಏನು ಕೋಡಾನ್ಸ್ ಇದೆ ಗೊತ್ತಾ? ಆ ಕೋಡಾನ್ಸ್ಗೆ ಆಂಟಿ ಕೋಡಾನ್. ಆಂಟಿ ಅಂದರೆ ಎಗೇನಿಸ್ಟ್ ತಾನೆ, ಎಗೇನಿಸ್ಟ್ ಅಂದರೆ ಕಾಂಪ್ಲಿಮೆಂಟಾರಿಟಿ ಅಂತ ಹೇಳ್ಬೋದು. ಸಪೋಸ್ mRNA ಮೇಲೆ GAG ಅಂತ ಒಂದು ಕೋಡಾನ್ ಇದೆ ಅಂತ ತಿಳ್ಕೊಳ್ಳಿ, ಓಕೆನಾ? ಈ GAG ಕೋಡ್ ವಿಚ್ ಅಮೈನೋ ಆಸಿಡ್ ನೋಡ್ರ ಆಗಿರ್ಬೇಕು ಅಂತ ಹೇಳಿದ್ದಾರೆ. ಗಾಗ್ ಈಸ್ ಗುಗ್ಗೆ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ದಾರೆ. ಗಾಗ್ ಅಂದ್ರೆ ಏನ? ಗ್ಲುಟಾಮಿಕ್ ಆಸಿಡ್. ಮತ್ತೆ ಸಪೋಸ್ ಈ TRNA ಬ್ರಿಂಗ್ಸ್ ದಟ್ ಒನ್, ವಿಚ್ ಒನ್? ಗ್ಲುಟಾಮಿಕ್ ಆಸಿಡ್ ಅಂತ ತಿಳ್ಕೊಳ್ಳಿ. ಗ್ಲುಟಾಮಿಕ್ ಆಸಿಡ್ನಿ ಈ ಅಮೈನೋ ಆಸಿಡ್ ಬ್ರಿಂಗ್ ಮಾಡಬೇಕಂದ್ರೆ ದೇ ಶುಡ್ ಹ್ಯಾವ್ ವಾಟ್ ಇಸ್ ಆಂಟಿಕೋಡಾನ್? CUC ಅಂತ ಆಂಟಿಕೋಡಾನ್ ಇರಬೇಕು. ಅರ್ಥ ಆಯ್ತಾ? ಸೊ ಕೋಡಾನ್ಸ್ ಆರ್ ಪ್ರೆಸೆಂಟ್ ಆನ್ mRNA, ಆಂಟಿಕೋಡಾನ್ಸ್ ಆನ್ TRNA. ದಟ್ಸ್ ವೈ ದೇ ಕ್ಯಾನ್ ಬ್ರಿಂಗ್ ದಟ್ ಆನ್ ದ ಅಮೈನೋ ಆಸಿಡ್. ಗಾಟ್ ದ ಕ್ಲಾರಿಟಿ ಸ್ಟೂಡೆಂಟ್ಸ್? ಮೇನ್ಲಿ ಆಂಟಿಕೋಡಾನ್ಸ್ ಪ್ರೆಸೆಂಟ್ ಆನ್ ದ ಆಂಟಿಕೋಡಾನ್ ಆರ್ಮ್. ಇದರಲ್ಲಿ 3 ಅನ್ಪೇಯ್ಡ್ ಬೇಸಿಸ್ ಆರ್ ದೇರ್. ಈ ಅನ್ಪೇಯ್ಡ್ ಬೇಸಿಸ್ನೇ ನಾವು ಆಂಟಿಕೋಡಾನ್ ಅಂತ ಕರಿತೀವಿ. ಈ ಆಂಟಿಕೋಡಾನ್ಸ್ ಆರ್ ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಟು ಕೋಡಾನ್ಸ್ ಆಫ್ mRNA. ಇಟ್ ಇಸ್ ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಟು ಕೋಡಾನ್ಸ್ ಆಫ್ mRNA. ಸ್ಟೂಡೆಂಟ್ಸ್, ಇದು ಆಂಟಿಕೋಡಾನ್ ಆರ್ಮ್ ಈಸ್ ಓವರ್. ಈಗ ನೆಕ್ಸ್ಟ್ ಒನ್ T-CIC ಆರ್ಮ್ ನೋಡಿ, ಆಕ್ಸೆಪ್ಟಾರ್ ಆರ್ಮ್ ಈಸ್ ಓವರ್. ನೆಕ್ಸ್ಟ್ ಒನ್ ಇಂದ ಆಂಟಿಕೋಡಾನ್ ಆರ್ಮ್ ಆಲ್ಸೋ ಓವರ್. ಈಗ ನೆಕ್ಸ್ಟ್ ರೈಟ್ ಸೈಡ್ಗೆ ಬರೋಣ. ರೈಟ್ ಸೈಡ್ನಲ್ಲಿ ನೋಡ್ತಾ ಇದ್ರ ಒನ್ ಆರ್ಮ್ ಈಸ್ ದೇರ್. ಈ ಆರ್ಮ್ಗೆ ಏನು ಹೆಸರಪ್ಪ? T-CIC ಆರ್ಮ್ ಅಂತ ಕರೀತಾರೆ. ಸೈಸಿ. ಯಾಕೆ ಸರ್ ಸೈ? ಅದೇನು ಸರ್ ಟಿ ಅಂತ ಹೊಡೆದಿರು? ಇದು RNA ಅಲ್ಲ ಸರ್, ಥೈಮಿನ್ ಹೆಂಗೆ ಬರುತ್ತೆ ಅಂತ ಕೆಲವೊಂದು ಸತಿ ಡೌಟ್ ಬರ್ಬೋದು. ಬಟ್ ಇವೆಲ್ಲ ಅನ್ಯೂಶುವಲ್ ಬೇಸಿಸ್. ಟಿ ಅಂದರೆ ಸೇಮ್ ಥೈಮಿಡಿನ್, ಮೇನ್ಲಿ ಥೈಮಿಡ್ ರಿಲೇಟೆಡ್ ನೈಟ್ರೋಜನ್ ಬೇಸಿಸ್. ಬಟ್ ಆಕ್ಚುಲಿ ಥೈಮಿನ್ ಇರಲ್ಲ RNA ನಲ್ಲಿ. ಬಟ್ ಈ TRNA ಮೇಲೆ ಮಾತ್ರ, ಈ T-CIC ಅವನಲ್ಲಿ ಮಾತ್ರ ಟಿ ಇದೆ. ಬಟ್ ಇಟ್ ಈಸ್ ಅನ್ಯೂಶುವಲ್ ನೈಟ್ರೋಜನ್ ಬೇಸ್ ಅಂತ ಕರಿತೀವಿ ಸ್ಟೂಡೆಂಟ್ಸ್, ಓಕೆನಾ? ಅನ್ಯೂಶುವಲ್ ನೈಟ್ರೋಜನ್ ಬೇಸ್. ಕ್ಲಿಯರಾ? ಓಕೆ. ಸೊ ಟಿ ಈಸ್ ಕಾಮನ್ಲಿ ಕಾಲ್ಡ್ ಥೈಮಿಡೈಲಿಕ್ ಆಸಿಡ್. ಕರೆಕ್ಟೇ, ಥೈಮಿಡೈಲಿಕ್ ಆಸಿಡ್. ಬಟ್ ಸೈ ಅಂದ್ರೆ ಏನು ಅಂದರೆ ಸೂಡೋಯೂರಿಡಿನ್. ದೀಸ್ ಟು ಆರ್ ಕಾಲ್ ಅನ್ಯೂಶುವಲ್ ನೈಟ್ರೋಜನ್ ಬೇಸಿಸ್. ಈ ಅನ್ಯೂಶುವಲ್ ನೈಟ್ರೋಜನ್ ಬೇಸಿಸ್ ಈಸ್ ಕಾಮನ್ಲಿ ಪ್ರೆಸೆಂಟ್ ಇನ್ ದ ವಿಚ್ ಆರ್ಮ್? T-CIC arm. Okay, and it helps to binding with which one? Ribosome ಜೊತೆಗೆ ಅಟ್ಯಾಚ್ ಆಗಬೇಕಂದ್ರೆ ಇದು ಯೂಸ್ ಆಗುತ್ತರ ಅಲ್ವಾ ಸ್ಟೂಡೆಂಟ್ಸ್? ಯಾಕಂದ್ರೆ TRNA ಬ್ರಿಂಗ್ಸ್ ಅಮೈನೋ ಆಸಿಡ್ ಎಲ್ಲಿವರೆಗೂ ತಗೊಂಡ್ಬರ್ತೀರಾ? ಓಕೆನಾ? ಸೊ TRNA ಬ್ರಿಂಗ್ಸ್ ಅಮೈನೋ ಆಸಿಡ್ to the which site under ribosomes. ಮತ್ತೆ ಇಲ್ಲೇ ribosomes ಇದೆ ಅಂದ್ರೆ how this TRNA can be find out ಅಂದ್ರೆ with the help of the which arm? T-CIC arm is find out that one the ribosome ಇದೆ ಅಂದ್ರೆ ribosomes ವರೆಗೆ ಬಂದು they can attached. ಈ ribosome ವರೆಗೆ ಬಂದು ಅಟ್ಯಾಚ್ ಆಗಬೇಕಂದ್ರೆ which part of the TRNA is very helpful? T-CIC arm is very helpful. ಕರೆಕ್ಟಾ? Because protein synthesis occur in the ribosome. That's why ribosomes ಗೆ ಅಟ್ಯಾಚ್ ಆಗಬೇಕಂದ್ರೆ ಇದು ಇಂಪಾರ್ಟೆಂಟ್. Got the clarity students? ಇದು T-CIC arm is there. T-CIC arm students, it is helpful for what? Binding with ribosomes. Again next one, the opposite to T-CIC arm, one more arm also there, that is called which arm? DHU arm. DHU ಅಂದ್ರೆ full form dihydroxyuridine. ಇದನ್ನು ಅನ್ಯೂಶುವಲ್ ಬೇಸ್ ಅಂತ ಕರೀತಾರೆ. ಅನ್ಯೂಶುವಲ್ ಅಂದ್ರೆ ಏನು? ಅಬ್ನಾರ್ಮಲ್ ಬೇಸ್ ಅಂತ ಹೇಳ್ಬೋದು, ಅದಕ್ಕೆ ಇದಕ್ಕೆ DHU arm ಅಂತ ಕರೀತಾರೆ. ಸೇಮ್ DHU arm ನಲ್ಲಿ ಆಲ್ಸೋ ಸ್ಟೆಮ್ ಇಸ್ ದೇರ್ ಅಂಡ್ ಲೂಪ್ ಆಲ್ಸೋ ದೇರ್. ಓಕೆನಾ? ಸೊ ಈ DHU arm ಸ್ಪೆಷಾಲಿಟಿ ಏನಂದ್ರೆ ಇಟ್ ಈಸ್ ಹೆಲ್ಪ್ಫುಲ್ ಫಾರ್ ಐಡೆಂಟಿಫಿಕೇಶನ್ ಆಫ್ ಅಮೈನೋ ಅಸೈಲ್ ಸಿಂಥಟೇಸ್ ಎಂಜೈಮ್. DHU arm, ಓಕೆನಾ? ಸೊ DHU arm ಬಗ್ಗೆ ಡಿಸ್ಕಸ್ ಮಾಡೋಣ. It is completely helps in identification or binding, anyone we can call binding to the enzyme, which enzyme andre, that is called amino acyl synthetase. So very important students. ಯಾಕಂದರೆ ಈ TRNA ಹೆಂಗೆ ಗೊತ್ತಾಗುತ್ತಾರೆ ಇಲ್ಲೇ ಅಮೈನೋ ಆಸಿಡ್ಸ್ ಇದೆ ಅಂತ? ಹೌ TRNA ಕ್ಯಾನ್ ಬಿ ಫೈಂಡ್ ಔಟ್ ಹಿಯರ್ ಓನ್ಲಿ ಅಮೈನೋ ಆಸಿಡ್ಸ್ ಆರ್ ದೇರ್? ಯಾಕಂದರೆ ಅಮೈನೋ ಆಸಿಡ್ ಜೊತೆಗೆ ಬೈಂಡ್ ಆಗಬೇಕಂದ್ರೆ ಫಸ್ಟ್ ಅಮೈನೋ ಆಸಿಡ್ ಎಲ್ಲಿದೆ ಅಂತ ತಿಳ್ಕೊಬೇಕು ತಾನೆ. ಅಮೈನೋ ಆಸಿಡ್ಸ್ ಎಲ್ಲಿದೆ ಅಂತ ತಿಳ್ಕೊಬೇಕಂದ್ರೆ ಫಸ್ಟ್ ದಿಸ್ ಶುಡ್ ಫೈಂಡ್ ಔಟ್ ದಿಸ್ ಇಸ್ ದ ಲೊಕೇಶನ್ ಆಫ್ ಅಮೈನೋ ಆಸಿಡ್. ಬಟ್ ಹೌ ದೇ ಕ್ಯಾನ್ ಫೈಂಡ್ ಔಟ್ ಅಂದರೆ ವಿತ್ ದ ಹೆಲ್ಪ್ ಆಫ್ ದ ವಿಚ್ ಆರ್ DHU. ಬಟ್ ಒನ್ ಸ್ಪೆಷಾಲಿಟಿ ಇಸ್ ದೇರ್. ವಾಟ್ ಇಸ್ ದ ಸ್ಪೆಷಾಲಿಟಿ ಮೀನ್ಸ್? Generally, protein synthesis time, amino acids are inactive. But before protein synthesis, all amino acids are inactive. Inactive amino acids can be activated by an enzyme. What is the name of the enzyme? Amino acyl synthetase. So, amino acyl synthetase enzyme is already released in the cytoplasm. Means what? All enzymes are become activate. So, all enzymes are activate andre. All amino acids are activate ಆಯ್ತಂದ್ರೆ ಎಸ್ ಆ ಆಕ್ಟಿವೇಟೆಡ್ ಅಮೈನೋ ಆಸಿಡ್ಸ್ ಅದಕ್ಕೆ ಈ ಎಂಜೈಮ್ ಎಲ್ಲಿದೋ ಆಟೋಮೆಟಿಕಲಿ ಓಕೆನಾ ವೇರ್ ಡಸ್ ಅಮೈನೋ ಆಸಿಡ್ ಸಿಂಥೇಸ್ ಎಂಜೈಮ್ ಇಸ್ ದೇರ್ ಆಟೋಮೆಟಿಕಲಿ ದೇರ್ ವಿಚ್ ಟೈಪ್ ಆಫ್ ಅಮೈನೋ ಆಸಿಡ್ಸ್ ಆರ್ ದೇರ್ activated amino acids are there. Are you observing students? Amino acid synthetase enzyme is helpful for activation of amino acids. Clear? Means where does this enzyme is present, automatically activated amino acids only present. That's why tRNA with the help of DHU arm they can find out the location of this enzyme. If they can find out the location of this enzyme means automatically tRNA can be find out the amino acid location also. Correct? Why? Because this enzyme always functioning that one activating the amino acid. Activating the amino acids. That's why E enzyme location find out madidandre automatically amino acids also located there itself only. Okay, it is helpful for identification or binding site to the which enzyme? Amino acid synthetase enzyme. What is the function of this enzyme? Amino acids. Got the clarity? Okay now? So protein synthesis, totally protein synthesis machinery in tRNA is very very important. Why? Because it is bring that one the amino acid. Why? Because amino acids are the raw material for protein synthesis. Amino acids are the raw material, but E raw materials is scatteredly present in the cytoplasm. That's why that amino acids are bring by this tRNA. That's why tRNA with the help of the T-size arm they can find out the location of ribosome. That's why they can bind with the ribosome. But how this tRNA can be find out that one here only amino acid is there means with the help of this enzyme. Correct? That's why E enzyme binding sites are present in the which site? DHU arm. DHU full form dihydroxyuridine. It is one of the unusual base is present in this arm. Here also two unusual bases are there. Okay, now that is called T-size. T means what? Thiamidylic acid. Psi means what? Pseudouridine. Clear students? Okay, now these are. You got one more arm is left. What is the name? This is called variable arm. This is called variable arm. But variable arm function is still unknown. It does not perform any function also. Does not perform any function. Got the clarity students? Okay now this is variable arm totally. Okay, five arms are present students. 5 arms: acceptor arm, anticodon arm, T-size arm, variable arm, last one in the DHU arm. These are the parts we can observe in the structure of which one? tRNA. Clear? Any tRNA also, okay? But tRNA secondary structure nalli it looks like a clover leaf, but in 3D picture 3-dimension picture nalli nodavaga inverted L shaped ಆಗಿರುತ್ತೆ. Clear? But in new textbook the diagram of tRNA they are given that one hanging thread ತರ ಕೊಟ್ಟಿದ್ರು ನೋಡಿ. ಊರಿ ಮಾಡ್ತಾರಲ್ಲ ಆ ಟೈಮ್ ನಲ್ಲಿ ಹೆಂಗೆ ಥ್ರೆಡ್ ಇರುತ್ತೋ ಹಂಗೆ ಕೊಟ್ಟಿದ್ರು ನೋಡಿ. Are you observing students? Okay now in your textbook they are given like this. tRNA ಅಂತ ಕೊಟ್ಟಿದ್ರು ನೋಡಿ. Correct? This is called 5 prime, this one is 3 prime. 3 prime again is which one is attached? Amino acid. But that diagram also don't leave students, because there also information is there. 2 amino acids they are given, 2 tRNAs they are given, you know. 1 tRNA is carries which amino acid? Tyrosine, correct? One tRNA is carrying which amino acid? Which one? Serine. Come on, what is the anticodon of the tRNA? Mainly tyrosine amino. Tyrosine carrying tRNA is having AUG. AUG is a anticodon, correct? AUG is the anticodon. Where it is present? tRNA. But AUG anticodon tRNA brings methionine amino acid? No, it is not bring methionine amino acid. It brings tyrosine amino acid. What about the serine anticodon? Serine anticodon, which one? Yes, UCA. Yeah, anticodon, UCA. Anticodon. It is carrying graph. Okay. So that is very, very important, friends. Don't leave any diagram in your NCRT nalli ಒಂದು diagram miss ಮಾಡಬೇಡಿ. Sana students, idura requirement ಮಾತ್ರ finish ಆಯಿತು. Plus yesterday class we discussed ribosomes also. Ribosomes we discussed. Ribosomes are commonly called naked organelles. Remember students, naked organelles. Ribosomes are made up of 2 components. Ribosomes are commonly made up of 2 components. I'm not asking about subunits. I'm asking about 2 components. Name out. rRNA is present and proteins. Come on, how many different type of proteins are there? 80 different type of protein. It is there in your NCRT. In second PUC textbook, in this molecular base of inheritance, 80 different type of amino acids are there in the structure of the protein. In the structure of the ribosome. In the structure of the ribosome, 80 different type of the proteins are there. Plus, which RNA? rRNA is present. Clear? Yesterday class we discussed. In prokaryotes, how many different type of rRNAs are there? 4. Eukaryotes are having 4 different types. Prokaryotes are having 3 different. Speak out what are the 4 different type of rRNAs in the eukaryotes. 28S, 5.8S, 5S, present in larger subunit. In the smaller subunit, 18S is present. Correct? In smaller subunit, 18S. I'm asking about only rRNA. rRNA only I'm asking. But what about prokaryotes? Prokaryotes are having, yes, how many different types of rRNA? 3 different type of rRNA. 23S, 5S, 16S. But 16S rRNA, where it is present? Yes. But one time question they are asked. Which rRNA of prokaryotes they can synthesize ribozyme? 23S rRNA, correct? 23S rRNA. Why? Because 23S rRNA present in smaller subunit or larger subunit. Same question if they will ask for eukaryotes, what is the answer you can give? 28S. Like that you can give the answer. Got the students? Okay, now? Today we can go for mechanism of translation, students. Mechanism of translation. Okay, na? So next, mechanism of translation. Mechanism of translation. Okay, students, in textbook they are given a mixing type of answer. Okay, so that's why I can separate and I can give. So totally how many stages are there in the mechanism of andre? 5 stages are there. 5 stages. But 2008 NEET nalli kottide question en gotta? Which one is the first step of translation? Which one? Activation of amino acids. Or it is also called, that is called charging of tRNA. Okay, na? That is about, okay? So that is called amino acylation. Amino acylation of tRNA or charging tRNA. Charging tRNA anta kirtara. Amino acyl tRNA, or it is also called charging tRNA. Second one in the, what about the second one? Andre, formation of mRNA ribosomal complex. mRNA ribosomal complex. Formation of mRNA ribosomal complex. Third one is called initiation of polypeptide chain. Initiation of polypeptide chain. Fourth one is called elongation of polypeptide chain. Elongation of polypeptide chain. And last one is called termination. Termination of polypeptide chain. 5 steps. 5 step nalli let us we have to discuss about first step. First step we can discuss. First of all, amino acylation of tRNA. First amino acids are there. Amino acids. It is in inactive state. The inactive state of amino acids can be activated by ATP plus one enzyme also there. Just before I said, amino acyl synthetase. So amino acyl synthetase plus ATP plus amino acid, both are bind agi, they will form amino acid AMP enzyme complex is formed. Plus PP. Next, AAAMP enzyme complex plus which one? That is called tRNA. Bind agi, they will form amino acyl tRNA or charged tRNA or charging tRNA anta karita. Plus which one? That is called that is called AMP plus enzyme. ಏನು ಸರ್ ಇದು ಅಮೈನೋ ಅಸೈಲೇಷನ್ ಆಫ್ tRNA ಅಂದರೆ ಏನು? ಏನಿಲ್ಲ, ಇವೆಲ್ಲ ಒಂದು ಸೈಟೋಪ್ಲಾಸಮ್ ಅಂತ ತಿಳ್ಕೊಡ. ಸೈಟೋಪ್ಲಾಸಮ್, ಈ ಸೈಟೋಪ್ಲಾಸಮ್ನಲ್ಲಿ ಇದೊಂದು ಅಮೈನೋ ಆಸಿಡ್, ಇದೊಂದು ಅಮೈನೋ ಆಸಿಡ್, ಇದೊಂದು ಅಮೈನೋ ಆಸಿಡ್. ಓಕೆನಾ? ಗೊತ್ತಾರ? ಎವ್ರಿ ಅಮೈನೋ ಆಸಿಡ್ ಸ್ಟ್ರಕ್ಚರ್ ವೆನ್ ಯು ಹ್ಯಾವ್ ಟು ಅಬ್ಸರ್ವ್ ದಟ್ ಒನ್ COOH is present at one side. Which one is there? Amino group is there. One side hydrogen, one side variable group is there. Remember? ಇದು ಅಮೈನೋ ಆಸಿಡ್ ಸ್ಟ್ರಕ್ಚರ್ ತಾನೆ. ಈ ಥರ ಮೆನಿ ಅಮೈನೋ ಆಸಿಡ್ಸ್ ಆರ್ ದೇರ್, ಆಲ್ ಆರ್ ಇನ್ಆಕ್ಟಿವ್. ಈ ಇನ್ಆಕ್ಟಿವೇಟೆಡ್ ಅಮೈನೋ ಆಸಿಡ್ని ಆಕ್ಟಿವೇಟ್ ಮಾಡೋಕ್ಕೆ ಏನು ಬಂದಿದೆ? ATP ಬಂದಿದ್ದೀರ. ಬಟ್ ATP ಏನಾಗಿತ್ತಂದರೆ ಬ್ರೇಕ್ ಡೌನ್ ಆಗಿ AMPಗೆ ಫಾಸ್ಫೇಟ್ಗೆ, ಹೌ ಫಾಸ್ಫೇಟ್? 2 ಫಾಸ್ಫೇಟ್ಗೆ ಬ್ರೇಕ್ ಡೌನ್ ಆಯಿತು. ಅವಾಗ ಏನು ರಿಲೀಸ್ ಆಯಿತು? ಎನರ್ಜಿ ರಿಲೀಸ್ ಆಯ್ತು. ATP hydrolysis ಆಗಿ they can convert it into which one? AMP and PI, they are releasing the which one? Energy. ಆ ಎನರ್ಜಿ they can activating the which one? Amino acid. Plus ATP ಇಂದ ಎನರ್ಜಿ ರಿಲೀಸ್ ಆಯಿತು, ಪ್ಲಸ್ ಅಮೈನೋ ಆಸಿಡ್ ಸಿಂಥೇಸ್ ಎಂಜೈಮ್ ಆಲ್ಸೋ ಆಕ್ಟಿವೇಶನ್, enzyme already is there. Okay, so amino acid in the presence of ATP, in the presence of amino acid synthetase, ಎರಡು bind ಆಗಿ, ಮೂರು bind ಆಗಿ, they will form a complex name out. What is the name of the complex? Amino acid. ಇದರ A ಅಂದ್ರೇನ? amino acid plus fall AMP. Just before I said, AMP also will be combined plus enzyme. What is the name of the enzyme? Amino acid synthase. ಈ ಮೂರು ಕಾಂಪ್ಲೆಕ್ಸ್ ಗೆ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ. ಪ್ಲಸ್ ವಿಚ್ ಒನ್ ಇಸ್ ರಿಲೀಸ್ಡ್? PPI ಅಂತ ರಿಲೀಸ್ ಆಗುತ್ತಾರೆ. P ಅಂದ್ರೇನು? ಫಾಸ್ಫೇಟ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ರಿಲೀಸ್ ಆಗ. ಹೌ ಮೆನಿ ಫಾಸ್ಫೇಟ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್? 2 ಫಾಸ್ಫೇಟ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ರಿಲೀಸ್ ಆಗುತ್ತೆ. ಅದಕ್ಕೆ PP ಅಂತ ಬರೆಯುತ್ತೆ. ಸ್ಟೂಡೆಂಟ್ಸ್, ಅಮೈನೋ ಆಸಿಡ್, ATP, ಅಮೈನೋ ಆಸಿಡ್ ಸಿಂಥೇಸ್ ಮೂರು ಮಿಕ್ಸ್ ಆಗುತ್ತೆ. ಮಿಕ್ಸ್ ಆಗಿ ದೇ ಕ್ಯಾನ್ ಫಾರ್ಮ್ AAAMP ಎಂಜೈಮ್ ಕಾಂಪ್ಲೆಕ್ಸ್. ಮತ್ತು ಇಮ್ಮಿಡಿಯೇಟ್ಲಿ AAAMP ಎಂಜೈಮ್ ಕಾಂಪ್ಲೆಕ್ಸ್ ನಲ್ಲಿ ಈ ಅಮೈನೋ ಆಸಿಡ್ ಆಲ್ರೆಡಿ, ಅಮೈನೋ ಆಸಿಡ್ ಅಂದ್ರೆ ಏನೇ? ಇಟ್ ಇಸ್ ಆಕ್ಟಿವೇಟೆಡ್. ಈ ಆಕ್ಟಿವೇಟೆಡ್ ಅಮೈನೋ ಆಸಿಡ್ ದೇ ಕ್ಯಾನ್ ಶಿಫ್ಟೆಡ್ ಟು ವಿಚ್ ಒನ್? tRNA ಮೇಲೆಗೆ ಶಿಫ್ಟ್ ಆಗುತ್ತೆ. ನಾವು ಆಲ್ರೆಡಿ ಇದು ಚಾರ್ಜ್ಡ್ tRNA ಅಂದರೆ activated amino acid. ಈ activated amino acid transfer to which one? tRNA. Then the tRNA is what we can call amino acid tRNA or charged tRNA. So that's why now this is about the one tRNA ಅಂತ ತಿಳ್ಕೊಡ. ಓಕೆನಾ? ನಾನು ಕ್ಲೋವರ್ ಲೀಫ್ ಮಾಡಲೇ ಡಯಾಗ್ರಾಮ್ ಡ್ರಾ ಮಾಡ್ತಿದ್ದೀರಾ. ಸೊ ದಿಸ್ ಇಸ್ ಆಕ್ಟಿವೇಟೆಡ್ tRNA. ಸೊ ಈ ಆಕ್ಟಿವೇಟೆಡ್ tRNA ಜೊತೆಗೆ ಅಮೈನೋ ಆಸಿಡ್ ಬೈಂಡ್ ಆಯಿತು. ದಿಸ್ ಇಸ್ ದ ಅಮೈನೋ ಆಸಿಡ್. Okay, but how amino acid can be find out by tRNA? With the help of their anticodons. Suppose if they want to bind with methionine amino acid, ಮಿಥಿಯೋನಿನ್ ಅಮೈನೋ ಆಸಿಡ್ ಜೊತೆಗೆ TRNA ಬೈಂಡ್ ಆಗಬೇಕಂದ್ರೆ ಕಂಪಲ್ಸರಿ ಮಿಥಿಯೋನಿನ್ ಮೇಲೆ ವಿಚ್ ಒನ್ ಇಸ್ ಆಂಟಿಕೋಡಾನ್ ಇರಬೇಕು, UAC ಆಂಟಿಕೋಡಾನ್ ಇರಬೇಕು. ಕರೆಕ್ಟಾ? ಸಪೋಸ್ CCC ಆಂಟಿಕೋಡಾನ್ ಇದೆ ಅಂತ ತಿಳ್ಕೊಳ್ಳಿ. CCE 3 C ಇದೆರಾ, ಓಕೆನಾ? ಇನ್ನೊಂದು TRNA ಇಸ್ ದೇರ್ ಅಂತ ತಿಳ್ಕೊಳ್ಳಿ. ಇನ್ನೊಂದು TRNA ಮೇಲೆ ಕಮಾಂದ, ಓಕೆನಾ? CCC ಅಂತ ಆಂಟಿಕೋಡಾನ್ ಇದೆ ಅಂತ ತಿಳ್ಕೊಳ್ಳಿ. ಯಸ್, it will be bring the prolina, it is bring the glycine amino acid ni think ಅಲ್ವಾ? ಯಾಕಂದ್ರೆ ಇದು ಆಂಟಿಕೋಡಾನ್ ಅಪ್ಪ. ಆಂಟಿಕೋಡಾನ್ ಪ್ರಕಾರ ಅಮೈನೋ ಆಸಿಡ್ ಇರಲ್ಲ, ಕೋಡಾನ್ ಪ್ರಕಾರ ಅಮೈನೋ ಆಸಿಡ್ ಇರುತ್ತೆ. ಇದು ತಿಳ್ಕೊಳ್ಳಿ. ಸಪೋಸ್ UUU ಆಂಟಿಕೋಡಾನ್ ಕಂಟೈನಿಂಗ್ TRNA brings which amino acid? UUU anticodon containing TRNA brings which amino acid? Which one? UUU containing which one? Correct? Correct? Yes, lysine. It is bring that one, the which one? Lysine. Lysine anta amino acid ni they can bring. Ekandre I am speaking anticodon, but don't think that one iga codon anta tilkobedi. Anticodon. ನಾನು ಆ್ಯಂಟಿಕೋಡಾನ್ ಹೇಳ್ತೀನಿ, ನೀವು ಕೋಡಾನ್ಗೆ ಥಿಂಕ್ ಮಾಡಿ ಆ ಕೋಡಾನ್ ವಿಚ್ ಅಮೈನೋ ಆಸಿಡ್ನಿ ಕೋಡ್ ಮಾಡುತ್ತೆ ಅಂತ ಐಡೆಂಟಿಫೈ ಮಾಡಿ. ಗಾಟ್ ಇಟ್? ಅದೇ ಸಪೋಸ್ ಒನ್ ಈಸ್ ಎ ದಟ್ ಈಸ್ ಕಾಲ್ GGG ಆ್ಯಂಟಿಕೋಡಾನ್ ಕಂಟೈನಿಂಗ್ TRNA brings which one? Proline and the amino acid they can brings like this. Again, got the clarity students? So anticodons present on TRNA. ಈ ಆ್ಯಂಟಿಕೋಡಾನ್ಸ್ ಪ್ರಕಾರ ಅಮೈನೋ ಆಸಿಡ್ ಬರಲ್ಲ. ಆ್ಯಂಟಿಕೋಡಾನ್ ಯಾವ ಕೋಡಾన్ని ಕೋಡ್ ಮಾಡುತ್ತೋ ಆ ಕೋಡಾನ್ ಪ್ರಕಾರನೇ ದೇ ಕ್ಯಾನ್ ಬ್ರಿಂಗ್ ದ ಅಮೈನೋ ಆಸಿಡ್. ಗಾಟ್ ದ ಕ್ಲಾರಿಟಿ? ಸೊ ಫಸ್ಟ್ ಒನ್ ಈಸ್ ಅವರ ಕ್ವಶನ್ ದರ್ ಆಸ್ಕ್ಡ್ ದಿಸ್ ಒನ್ only in 2018 or 19 or maybe. Okay na, the first step in translation ಅಂದ್ರೆ ಇದೆರಾ, ಚಾರ್ಜಿಂಗ್ ಆಫ್ TRNA or amino acylation of TRNA. So activated amino acids all are bind. Activated amino acids bind with whom? TRNA. Then they will form amino acyl TRNA. Finished. First step is finished. Again I want to explain which enzyme is very helpful for activation of amino acid. Yes, important. Where does it located? Cytoplasm ra. Cytoplasm e. ಯಾಕಂದ್ರೆ ಟೋಟಲ್ ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಆಗೋದು ಎಲ್ಲಿ? ಸೈಟೋಪ್ಲಾಸಮೇ. ಇದು ಪ್ರೋಕ್ಯಾರಿಯೋಟ್ ಆಗ್ಬೋದು, ಯೂಕ್ಯಾರಿಯೋಟ್ ಆಗ್ಬೋದು. Either in prokaryotes or eukaryotes, it is occurring in cytoplasm. The second step is called formation of mRNA ribosomal complex. You already know. Yes. Generally, gene express avaga compulsory gene can be synthesized protein. One gene express aagta ide andre first compulsory they can synthesize what? Protein. But how many levels are there in the gene expression in eukaryotes? 4 levels. What are the 4 levels? Means first one, transcription level. By the transcription they can synthesize hnRNA. They can synthesize. hnRNA undergoes processing. That's why the second level is processing of hnRNA. After the processing, hnRNA become mRNA. Correct? mRNA. But transcription, processing both are occur inside the nucleus. But after the processing, mRNA cannot stay in the nucleus. It can migrate from nucleus into cytoplasm. From nucleus it is enter into which one? Cytoplasm. Cytoplasm ge enter agi, then they can bind with whom? Okay na? Yes. What about the third one? Means that is called, yes Anna. First two are called, first of all, transcription. After the transcription in the processing. What about the third one? The migration meaning movement. The movement of mRNA from nucleus into cytoplasm. That is about the third step. Fourth step in the translation. By the translation only they can synthesize protein. This is gene expression nalli 4 stages. But what about in prokaryotes? Prokaryotes nalli how many levels are there? 2 levels. What is the name? Transcription level and translation level. There is no processing and there is no migration also. Correct? Why? Because there is no nuclear membrane. Nuclear membrane is not present. Both transcription, translation, both are occurring and both are coupled also. Coupled means what? If the transcription complete agalla, translation should agutte anta helidalla. Okay, now before completion of transcription only translation will be started. That's why both are coupled process in the prokaryotes. But eukaryotes not like that. Why? Because the segregation is there, separation is there between the nucleus and cytoplasm. But translation in both prokaryotes and eukaryotes occur in cytoplasm only. Clear? Now, first step, next one is mRNA ribosomal complex. This is called mRNA, students, mRNA. Already we discussed mRNA structure. mRNA first part what? What is the name of the first part of the mRNA? Leader. Yes or no? This is called the mass leader region or cap region. This is called what? Cap region. Cap region is always present near to which one? 5 prime. Come on now. What is the name of the group is present here? Methylated guanosine triphosphate is present in the 5 prime. Are they will form the cap or not? Clear? After the cap region, come on, the next one is UTR. UTR meaning untranslated region. Upper side, which one is there? Initiator. Okay, now what is the name? Initiator. Initiator region is there. Initiator region. Come on, what is the codon? AUG. 99% AUG is the initiator codon, but rarely GUG also codon. GUG also initiator codon anta helide, but GUG code well. Clear? After the initiator region, next region is called coding range. Coding range. After the coding range, come on, what is the name? Terminator region. Terminator region. Terminator region nalli, 3 codons nalli, any one of the codon is present. Students, either UAA, UAG, UG. After that one, come on, the next region is called UTR2. UTR, untranslated region 2 is there. And last one, which one? Tail. This is called tail. Tail is present in the which polarity? 3 prime. Clear, students? So UTR present before initiator and after terminator. Correct? Before initiator and after terminator. And that's why it is called UTR1. This is called UTR2. UTR2 anta karita. Clear? Now this mRNA, processed mRNA. But no processing in the prokaryote. That one you have to remember. Okay? Now this mRNA enter into cytoplasm. Then they can first bind with the smaller subunit of ribosome. Smaller subunit of ribosome. Smaller subunit of ribosome. Okay? The first they can bind with the smaller subunit of ribosome. For example, now we are discussing the mechanism of translation in prokaryotes anta tilkoli. Prokaryotes anta tilkoli. Avaga, which subunit first will come and bind here? Which subunit? Prokaryotes nalli what type of ribosome? 70S. Correct? 70S nalli smaller subunit? 30S. That's why first 30S ribosome or 30S smaller subunit binds. After that one, larger subunit will become and bind here like this. Larger subunit will become and bind. Clear, students? Then they will form a complex. What is the name of this complex? mRNA ribosomal complex. mRNA, this is mRNA or not? This is mRNA ribosomal complex. This is the second stage in the translation. mRNA ribosomal complex. mRNA ribosomal complex. First mRNA can bind with the smaller, later it is followed by larger. So what is the larger subunit in the prokaryotic ribosome? 50S. Then 50S can be bind. But 50S subunit andre larger subunit is having 3 sites. Can you speak out what the name of the 3 sites are there? Yes, what is the name of the sites? A site, P site, E site. Correct? EPA anta karitara. Simple agi ilbekana. First amino acid containing tRNA will be comes to which site? P site. Except first tRNA, remaining all tRNA first they will come to A site, later it is moved to P site. This is very important. Andre, what is the name of the tRNA? First come to the, what is the name? Andre, which amino acid containing tRNA? It will becomes to first P site. Which one? Methionine, no doubt at all. Methionine containing tRNA comes to P site only, not A site. Clear, students? This is very, very important. Okay, next, once these tRNA enter into the E site, they can leave the, once the tRNA, it will be enter into the P in the E site, enter aitondre, they can stay in the ribosome, they can move out of the ribosome, Kelsa enida. When they are move out of the ribosome, come on, they are leaving their amino acid, along with the amino acid they are leaving their amino acid. Only they can move out of the ribosome. This is important. Andre always ribosomes are giving the site for only 2 tRNAs only. Always ribosomes are giving the site for 2 tRNAs only. 1 tRNA can be bind to A site, 1 tRNA can be bind to B site. Suppose if any tRNA enter into the E site, means they can leave. They can leave that one, the ribosome. This point important, students. ಮತ್ತೆ ನಾನು ರಿಪೀಟ್ ಮಾಡಲ್ಲ, ಈ ಸೈಡ್ ಬರುತ್ತೆ ಅಂತ ಹೇಳಲ್ಲ, ಬಟ್ ನೀವೇ ತಿಳ್ಕೊಳ್ಳಿ. ಓಕೆ, ಎಸ್. ಸೆಕೆಂಡ್ ಸ್ಟೆಪ್ ಆಲ್ಸೋ ಓವರ್. ಈಗ ಥರ್ಡ್ ಸ್ಟೆಪ್ ಇನಿಷಿಯೇಷನ್ ಆಫ್ ಪಾಲಿಪೆಪ್ಟೈಡ್ ಚೈನ್. Okay, what is mean by initiation andre? So that is called amino acyl tRNA. Amino acyl tRNA comes and binds to P site. Amino acyl tRNA comes and binds to P site. It initiates transcription. It initiate translation. Amino acyl tRNA ಬಂತು P site ನಲ್ಲಿ ಅಟ್ಯಾಚ್ ಆಯಿತು ಅಂದರೆ ಎಸ್. Speak out which one is the first amino acyl tRNA. That is called met tRNA ಅಂತ ಕರೀತಾರೆ. Meaning what is meant by met? Methionine tRNA ಅಂತ ಕರೀತಾರೆ. And very important, when the largest subunit is completely comes and attached, are you observing AUG? Now it is present opposite to the which site of ribosome? AUG now present opposite to the which site of ribosome? P site. ಅದಕ್ಕೆ first amino acyl tRNA will be come to P site, not to A site. Why? This reason. This is the reason. Got the clarity, students? Okay? Yes. So amino acyl tRNA will be comes to P site and initiate translation. Okay? Next, amino acyl or second amino acyl tRNA comes and binds to which one? A site or P site? A site of ribosomes. Next, first amino acid amino group, carboxylic group, comma, second amino acid amino group between hydrogen bond is formed. Okay na? First amino acid carboxylic group, idu highlight ಮಾಡ್ಕೊಳ್ಳಿ students. Second amino acid amino group between hydrogen bond is formed. Peptide bond is formed. By which enzyme? Ribozyme enzyme. ಇದರ ಇನಿಷಿಯೇಷನ್ ಅಂತ ಕರೀತಾರೆ. ಇನಿಷಿಯೇಷನ್ ಆಯಿತು. ಏನು ಇನಿಷಿಯೇಷನ್? ಪಾಲಿಪೆಪ್ಟೈಡ್ ಚೈನ್ ಇನಿಷಿಯೇಷನ್. ಅದಕ್ಕೆ ಡಯಾಗ್ರಾಮ್ ಐ ಆಮ್ ಗಿವಿಂಗ್ ದ ಸೇಮ್. ವಾಟ್ ಈಸ್ ದ ಡಯಾಗ್ರಾಮ್ ಪ್ರಿವಿಯಸ್ಲಿ ಐ ಡ್ರಾನ್? ಸೇಮ್ ಡಯಾಗ್ರಾಮ್ ಐ ಆಮ್ ಡ್ರಾಯಿಂಗ್, ಸ್ಟೂಡೆಂಟ್ಸ್. ದಿಸ್ ಅಬೌಟ್ ದ ಸೇಮ್ ಡಯಾಗ್ರಾಮ್. ಇದು 5' polarity and this one is called which prime now? 3' polarity. Okay, so 5' polarity nalli already which one is attached now? This one is called ribosomes are attached. Okay, ribosomes. This is about the which site? P site. This is about the A site and it is A site. Now first amino acyl synthetase, first amino acyl tRNA, okay, will becomes and bind to the which region? P site. Next, what is the anticodon of the tRNA? This tRNA anta tilkodi. ನಿಮ್ಮ ಅರ್ಥಕ್ಕೋಸ್ಕರ ಈ ಥರ ಕ್ಲೋವರ್ ಲೀಫ್ ಮಾಡಲ್ನೇ ಐ ಕಂಪ್ಲೀಟೆಡ್ ಡ್ರಾಯಿಂಗ್ ಇನ್ ದ ಫಾರ್ಮ್ ಆಫ್ ಬಾಕ್ಸ್. ಒಂದು ಬಾಕ್ಸ್ ಥರ ಬರ್ತಾ ಇದ್ದೀರಾ TRNA. ಈ TRNA ಮೇಲೆ ವಿಚ್ ಆ್ಯಂಟಿಕೋಡಾನ್? UAC ಆ್ಯಂಟಿಕೋಡಾನ್ ಇದ್ದರೆ ಕಮಾಂಡ ಆ್ಯಂಡ್ ವಾಟ್ ಇಟ್ ಕ್ಯಾರೀಸ್? ಮಿಥಿಯೋನಿನ್. ನೆಕ್ಸ್ಟ್ ಆಫ್ಟರ್ ದ AUG ಸೆಕೆಂಡ್ ಕೋಡಾನ್ ಆಲ್ಸೋ ಪ್ರೆಸೆಂಟಾ. ಈ ಸೆಕೆಂಡ್ ಕೋಡಾನ್ ಏನಂತ ಬರ್ತಾ ಇದ್ದೀರಂದರೆ Yes, ಓಕೆನಾ GAG ಅಂತ ಬರ್ತಾ ಇದೆ ನಾವು ಸೆಕೆಂಡ್ ಕೋಡಾನ್ ಏನಂದ್ರೆ GAG ಅಂತ ಬರ್ತಾ ಇದೆ ಓಕೆ so now which tRNA will becomes here come on what is the anticodon containing tRNA will becomes to A site CUC containing anticodon tRNA brings which amino acid which one glutamic acid okay students here this is about the one of the amino acid can I write like this one COOH NH2 H this is about the first amino acid second amino acid can I write like this one NH2 can I write like this itara bariboda second this is about idondu amino acid students okay amino acid nalli what are the groups are there amino group is there carboxylic group is there same here also amino group is there carboxylic group is there so totally how many amino acids are there here 2 amino acid first amino acid is there in the which site P site second amino acid in A site first amino acid carboxylic group second amino acid amino group between come on which bond is formation peptide bond formation so when the peptide bond is formation is occur andre yes polypeptide chain is initiated occurs peptide bond formation ಆಯಿತು ಅಂದರೆ polypeptide chain is initiated ಅಂತ ಅರ್ಥ ಮತ್ತೆ ಈ peptide bond formation by come on which enzyme ribozyme enzyme is helpful for formation of the peptide bond students ribozyme is helpful for formation of the peptide bond between amino acids this is called peptide bond ಅಂತ ಕರೀತಾರೆ peptide bond speak out students peptide bond is present between smaller subunit or larger subunit okay now they can form the between methionine and glutamic acid according to this diagram methionine and glutamic acid but in the theory concept I am not written methionine okay so glutamic acid I am not written just first amino acid second amino acid first amino acid carboxylic group second amino acid amino group between peptide bond is formed by ribozyme enzyme here friends okay so initiation of polypeptide chain also over students see the next one elongation of polypeptide chain ಈ elongation of polypeptide chain ಯಾವಾಗ ಆಗುತ್ತಂದ್ರೆ ribosome move one codon to another codon or next codon ribosome move one codon to next codon on mRNA from 5' to 3' polarity 5' to 3' polarity okay so the movement of ribosomes on mRNA movement of ribosome on mRNA causes elongation of what polypeptide chain elongation ಆಗುತ್ತೆ how this elongation is occurs andre okay then we can discuss in the next diagram ನಲ್ಲಿ ನಾನು explain ಮಾಡ್ತು ಹೋಗ್ತಾ ಇದೀನಿ okay is it okay same mRNA I'm completely drawing here 5' to 3' polarity now ribosomes are moving from which polarity now 5' to 3' polarity ಸ್ವಲ್ಪ ಮೂವ್ ಆಯಿತು ಅಂತ ತಿಳ್ಕೊಳ್ಳಿ ಸ್ವಲ್ಪ ಮೂವ್ ಆಯಿತು ಅಂದರೆ ಇದೇ ಡಯಾಗ್ರಾಮ್ನಲ್ಲಿ ಸ್ವಲ್ಪ ಡಾಟ್ಸ್ ಮಾಡಿ ಹೇಳ್ತಾ ಇದ್ದೀರಾ ಸ್ಟೂಡೆಂಟ್ಸ್ ಓಕೆ ಸೊ ನಾವು ದಿಸ್ ಈಸ್ ಅಬೌಟ್ ಮೂವ್ಮೆಂಟ್ ಆಫ್ ರೈಬೋಸೋಮ್ಸ್ ಲೈಕ್ ದಿಸ್ ರೈಬೋಸೋಮ್ಸ್ ನೋಡಿ ಈ ಥರ ಸ್ವಲ್ಪ ಮೂವ್ ಆಯಿತು ಇದೇ ಡಯಾಗ್ರಾಮ್ ಹಿಯರ್ ಐ ಆಮ್ ಡ್ರಾಯಿಂಗ್ ಸೊ ದಿಸ್ ಅಬೌಟ್ ದ ರೈಬೋಸೋಮ್ಸ್ ಸ್ಮಾಲರ್ ಸಬ್ ಯೂನಿಟ್ ಆ್ಯಂಡ್ ಲಾರ್ಜರ್ ಸಬ್ ಯೂನಿಟ್ ಈ ಥರ ರೈಬೋಸೋಮ್ಸ್ ವೆನ್ ದ ಮೂವ್ಮೆಂಟ್ ಈಸ್ ಅಕ್ಕರ್ಸ್ ವಾಟ್ ವಿಲ್ ಬಿ ಹ್ಯಾಪನ್ ಪ್ರೀವಿಯಸ್ಲಿ ದಿಸ್ ಇಸ್ ಕಾಲ್ ವಿಚ್ ಸೈಟ್ ಅಂತ ಹೇಳ್ಬೋದು ವಾಟ್ ಇಸ್ ದ ನೇಮ್ ಆಫ್ ದಿಸ್ ಸೈಟ್ yes okay na so more sites bariyana okay na so more sites barta idini this is about what is the name of this site E site and what is the name of this site A site according to this diagram previously from A site which tRNA is there glutamic acid containing tRNA now glutamic acid tRNA now it will be enter into the which site P site okay na idu tRNA ra ee tRNA mele which one na glutamic acid between before that which one is there methionine is there correct okay Before that, methionine is there. Before that, methionine is already there. So that's why here what is happened means, okay, first amino acid TRNA, okay, now leave their amino acid and moves out of the ribosome through exit site in that they can move to outside. Through exit site, first amino acid containing TRNA, okay, leave their amino acid and moves out of the ribosomes. Move out of the ribosome andre, you see methionine containing TRNA enagutte, they can move out. Come on, idu TRNA, TRNA mele, come on, what is the codon sequence, what is the anticodon sequence is there? Methionine TRNA. Methionine TRNA mele, what is the name of the sequence? UAC. Alva, UAC anticodon containing TRNA is completely leaving from the, which one? Leaving from the ribosomes. So now that place is occupied by which amino acid containing TRNA? Glutamic acid containing. So here AUG codons illidirappa. Igo illi ಏನು codon sequence idara illi according to that diagram GAG ಅಂತ sequence ಇದೆ. Anticodon ಯಾವುದು? CUC. ಈಗ ಇನ್ನೊಂದು codon ಇದೆರಾ, ಅದೇ CCC ಅಂತ ಕರೆಯೋಣ. Yes, ಓಕೆನಾ? ಮತ್ತೆ ಈ CCC codon ಬರೆಯಕ್ಕೆ ಕಮಾಂಡ one more amino acid, one more TRNA is ready. ಆ TRNA ಮೇಲೆ ಕಮಾಂಡ what is the name of the codon? GGG. ಇದು TRNA, ಕರೆಕ್ಟಾ? ಈ TRNA ಮೇಲೆ ಕಮಾಂಡ which one is there? Proline is there. Ego, e proline containing TRNA will becomes here. Like this process will be going till up to the end of the which one? 3' polarity. 3' polarity ವರೆಗೂ ಇದು ಕಂಟಿನ್ಯೂ ಆಗುತ್ತರ. ಒಂದು ಬರುತ್ತೆ ಒಂದು ಹೋಗುತ್ತರ. Okay now. First one TRNA will be comes to here means what? What is the first P site nallide TRNA ಇದೆ ಅಲ್ವಾ? They are completely enter into the E site. E site ಗೆ enter ಆಯ್ತು ಅಂದ್ರೆ they can exit. Next A site ಖಾಲಿ ಆಗುತ್ತೆ. A site ಖಾಲಿ ಆಯ್ತು ಅಂದ್ರೆ next. Then ಮತ್ತೆ ribosome is move towards forward or move forward. If ribosome is move forward means ಮತ್ತೆ ಏನಾಗುತ್ತೆ? P site ನಿಲ್ಲಿದೆ E site ಗೆ enter ಆಗುತ್ತೆ. A site ನಿಲ್ಲಿದೆ P site ಗೆ enter ಆಗುತ್ತೆ. ಮತ್ತೆ which site is vacant? A site is vacant. Matte one more TRNA will come. Like this process will be continued till the terminator region ಬಂದುವರೆಗೂ ಇದು ಕಂಟಿನ್ಯೂ ಆಗುತ್ತಾ. ಇದೇ elongation ಅಂತ ಕರೀತೀವಿ. Got the clarity, students? Again, how to explain? No. Last one is called which one now? Termination. The last one is called, fifth one is called termination of polypeptide chain. Termination of polypeptide chain. So termination is commonly depend on, okay now, termination is depend on, yes, occurrence of which codon? Stop to codons or terminator codons. Ena terminator codons? Yes. Terminator codon. Anyone come to opposite to which site? Opposite to A site of ribosome, automatically polypeptide chain synthesis is stopped. Plus, very important, terminator factors are there. Or it is also called releasing factors. Releasing factors separates what? Okay, polypeptide from ribosomes. It is completely. This is terminator. You know, same diagram I am drawing. Okay, na? This is 5' to 3' polarity of ribosome, mRNA. ನಾವು ರೈಬೋಸೋಮ್ ಎಲ್ಲಿವರೆಗೂ ರೀಚ್ ಆಯ್ತು ಅಂದರೆ ಕಮಾಂಡ್ ಆಗಿ ಈ ರೀಜನಲ್ಲಿ ವಿಚ್ ರೀಜನ್ ಅಂತ ಕರಿತೀವಿ, ಟರ್ಮಿನೇಟರ್ ಅಂತ ಕರಿತೀವಿ. ಟರ್ಮಿನೇಟರ್ನಲ್ಲಿ ಯಾವ ಸೀಕ್ವೆನ್ಸ್ ಇದೆಪ್ಪ, UA ಆಗ್ಬೋದು, UAG ಆಗ್ಬೋದು, UG ಆಗ್ಬೋದು, ಎನಿ ಒನ್ ಕೋಡಾನ್. ಈ ಮೂರ್ನಲ್ಲಿ ಎನಿ ಒನ್ ಕೋಡಾನ್ ಇದ್ದರೆ ಏನಾಗುತ್ತೆ? ಓಕೆ, ಇಸಿ ನಾವು ದಿಸ್ ಇಸ್ ಅಬೌಟ್ ದ ರೈಬೋಸೋಮ್. ಈಗ ನಿಯರ್ ಟು ಟರ್ಮಿನೇಟರ್ ಕೋಡಾನ್ವರೆಗೆ ಬಂದಿದ್ದೀರಾ. ಈಗ ದಿಸ್ ಇಸ್ ಅಬೌಟ್ ದ P site, this is called A site, this is called which one? E site. ಸೊ ನಾವು ಇದು A site ಅಲ್ವಾ, ಮತ್ತೆ ನಾನು ಇಲ್ಲಿ ಏನು ಮರೆಯಲ್ಲ, ಓಕೆನಾ. ಬಟ್ A site ಆಪೋಸಿಟ್ ಯಾವುದು ಬಂದಿದ್ರೆ UAA ಅಂತ ಕೋಡಾನ್ ಬಂದಿದೆ. ಆಲ್ರೆಡಿ P ಸೈಟ್ನಲ್ಲಿ ಓಕೆನಾ, ಆಲ್ರೆಡಿ TRNA ಇಸ್ ದೇರ್. ಈ TRNA ಮೇಲೆ ಕಮಾಂಡ ವಿಚ್ ಸೀಕ್ವೆನ್ಸ್ ಇಸ್ ದೇರಾ, ದಿಸ್ ಇಸ್ ಅಬೌಟ್ ಪಾಲಿಪೆಪ್ಟೈಡ್ ಸೀಕ್ವೆನ್ಸ್ ಆಲ್ರೆಡಿ ಇಸ್ ದೇರ್. ಇದೆಲ್ಲ ಎಲಾಂಗೇಷನ್ ಆಯಿತು ಅಂತ ತಿಳ್ಕೊಬೋದಲ್ಲ. ಇವೆಲ್ಲ ವಾಟ್ ಇಸ್ ದ ನೇಮ್ ಆಫ್ ದಿಸ್ ಒನ್? ಅಮೈನೋ ಆಸಿಡ್ಸ್. ಇವೆಲ್ಲ ಅಮೈನೋ ಆಸಿಡ್ ಅಂತಲೇ ಬರಿತಾ ಇದ್ದ. ನಾನು ಐ ಆಮ್ ನಾಟ್ ಸ್ಪೆಸಿಫೈಯಿಂಗ್. ದಿಸ್ ಇಸ್ ಅಬೌಟ್ ದ ಅಮೈನೋ ಆಸಿಡ್. ಕ್ಲಿಯರ್ ಸ್ಟೂಡೆಂಟ್ಸ್? ಸೊ ಅಮೈನೋ ಆಸಿಡ್ or polypeptide chain elongation is depend upon the movement of ribosome on mRNA. Clear? Next, mRNA or ribosomes are reached upon the 3' polarity of mRNA. Okay now, then what will be happened andre termination is occurs. But the termination depend upon occurrence of terminator codon or stop codons. Name out these three. Any one of the codon comes opposite to A site, this is called A site, opposite to A site, any one of the codon, then what will be happen? Polypeptide chain synthesis is stopped. Why? Because terminator codon or stop codon does not code any amino acid. That's why it is also having any TRNA. Okay, any TRNA is there? No TRNA is there. Okay, na? So that's why no amino acid is brings. Clear? That's why protein synthesis is stopped. When the protein synthesis stopped, come on, the polypeptide chain is segregate. Correct? Ribosome also segregate. Ribosome also separate. Got the clarity, students? Okay, na. Idu total translation concept or translation. Era newly synthesized protein, what we can call primary structure of the protein. Then primary structure of the protein later changed into secondary become tertiary become quaternary. But in these 4 stages, come on, the enzymes are present in which state? Enzymes are present in which state? Andre tertiary stage ante irbeku. Correct? Enzymes all are majoritarily 99% of the enzymes are made up of protein alva. Enzymes all are protein, no? But these proteins are present in which state? Tertiary.